Hello guys, this is Vineet and welcome to the molecular biology series, chapter number 4. So today I'll be discussing about the structure and conformations of the DNA. So when we talk about the DNA, this is a anionic biopolymer. So that means a negatively charged long polymer chain which contains the nucleotides as the monomers. Okay, so the nucleotides which are present in the DNA are the deoxyforms, adenine deoxyribonucleotide, thymine deoxyribonucleotide, guanine deoxyribonucleotide, and the cytosine deoxyribonucleotide. These are the major nucleotides, deoxyribonucleotides, which are present in the DNA. And the chromosomal DNA consists of very long DNA molecules, approximately weighing about 1.6 into 10 to the power of 6 dalton to 2 into 10 to the power of 9 daltons. Okay, and each DNA polymer. So when we talk about the polynucleotide chain, so this polymer is about 10 to the power of 10 deoxyribonucleotides. Okay, so this simple long polymer chain contains the 10 to the power of 10 deoxyribonucleotides. Okay, and these are the normal bases which are normal nucleotides which are present in the DNA. So this is the primary structure of the DNA, and this is how the structure of the DNA is formed, inside the cells. And these nitrogenous bases are linked by the special type of bond called as a phosphodiester linkage. Okay, so and this runs from 3' to 5' direction. And here what you are seeing is the single strand of the DNA, and when you have another strand parallelly or in anti-parallel direction, okay, so that makes up the double helical structure. Okay, now how does this phosphodiester linkage is formed between the nucleotides? Okay, so we have here the adenine molecule. So this is the adenine nucleotide, and we have the nitrogen pentose sugar over here, which contains a phosphate group, this one. So the phosphate group is present on the 5th carbon. Okay, and this phosphate molecule which is present on the 5th carbon will get bonded to the 3rd carbon of the above nucleotide. Okay, so the above nucleotide is what? Cytosine nucleotide. Okay, so this phosphate molecule which is present on the 5th carbon will now bond with the 3rd carbon hydroxyl group of the hydroxyl group of the 3rd carbon of cytosine nucleotide. Okay, so this kind of reaction is also known as a condensation reaction and involves elimination of a water molecule and forms a ester bond. Okay, so this bond is known as the ester bond, and down the phosphate atom we also have another ester bond. Okay, so since this bond has two ester bonds, we call this as the phosphodiester bond. Okay, okay, so phosphodiester bond. Similarly, the cytosine also contains the phosphate group on the 5th carbon. Okay, and same phosphate group now get bonded to the hydroxyl group of the 3rd carbon of the above cytosine molecule. So this cytosine also contains the sugar molecule, and this phosphate molecule will now gets bonded to the hydroxyl group of 3rd carbon. Okay, and it also involves the condensation reaction and eliminates the water molecule from during the bond formation and forms an ester bond. Okay, here also we have two ester bonds up and down, so we call this as the phosphodiester bonds. Okay, so entire polynucleotide chain is formed by the phosphodiester linkages, and these phosphodiester linkages generally act as the backbone for the polynucleotide chain. Okay, and so this is one nucleotide chain. So if we have another nucleotide which runs in anti-parallel direction like this, okay, and this chain will be going in this direction. Okay, so one strand runs in the 5' to 3' direction in this way, another strand runs in the anti-parallelly 5' to 3' direction. Okay, so this anti-parallel orientation will give the double helical nature of the DNA. Okay, so like double stranded structure of the DNA. Okay, so whenever you take any single polynucleotide chain, it generally ends with a 3' hydroxyl group and 5' phosphate groups. Okay, in the end of the any polynucleotide chain, you will have a 3' hydroxyl group over here. Okay, here in this position, and at the end of the chain, at the 5 prime end, you will have the phosphate molecule. Okay, so this is the these are the ends of the any polynucleotide chain. Okay, so similarly you also have another anti-parallel nucleotide chain. Okay, so in the which is running in the opposite direction. Okay, so that chain also has the 5 prime for phosphate group in the end, and also has the 3 prime hydroxyl group in the end. Okay, so this is how the phosphodiester linkage is formed in the nucleotide polynucleotide chain. And that is how the nucleotide bases are linked to one another. Okay, so this is about the actual formation of the single nucleotide strand, and I've said you already. So the end of the strand which bears the 5 prime phosphate group without phosphodiester linkage is called the 5 prime end, and the opposite end bears a free 3 prime hydroxyl group. Okay, or 3 phosphate group. It is called as the 3 prime end. Okay, so you have to remember the ends of the polynucleotide chain or strand. Okay, now the primary structure is the number and sequence of different deoxyribonucleotides in strands joined by the phosphodiester linkages, which is also called as the backbone of the DNA strand. Okay, so this backbone of the primary structure, okay, uh, make the strand to have a linear structure. Okay, so without any bends or kinks. Okay, without any change in the angle or anything. Okay, so this phosphodiester linkage makes the DNA strand as a linear strand. Okay, now comes with the secondary structure of the DNA. So what we have discussed is the primary structure, which involves the simple nucleotides and formation of the phosphodiester linkages. Okay, now the double helical structure, which is also regarded as the secondary structure of the DNA and proposed by the James Watson and Francis Crick, and also Rosalind Franklin. Okay, so James Watson and Francis Crick have actually figured out the structure of the DNA and constructed a double helical structure of the DNA and represented it in front of the committee, in front of a great scientist. Okay, and explain the properties of this double helical structure of the DNA. So we will also see the properties of the double helical structure of DNA, what Watson and Crick has proposed. Okay, so its diameter is about 2 nanometers, and each turn of the helix is about 3.4 nanometers, 34 angstroms. Okay, and the two strands are antiparallel, as I have said you. They run in opposite directions. Okay, one strand runs in the 5 prime to 3 prime direction, while the other strand runs in the 3 prime to 5 prime direction. Okay, so this kind of antiparallel direction gives the complementarity, but not the identity. Okay, so that means these two chains in the double helical structure of the DNA are not identical. Okay, so they are complementary. Okay, complementary is different from the identical chains. Okay, now if we talk about the history of this secondary structure of the DNA, actually Rosalind Franklin, she was the first scientist to figure out the X-ray crystallographic structure of the DNA, and she was able to print the structure of this double helical DNA as a X-ray crystallographic image. Okay, and later this Watson and Crick, they figured out a lot of unpublished work of this Rosalind Franklin, and they took out the unpublished work and they published on their names. Okay, so lot of properties of the DNA were already studied by the Rosalind Franklin, but unfortunately she was unable to publish them. Okay, and Watson and Crick took their chance and got the some sort of result from the Franklin works, and they published in their own way. Okay, so these some major properties of the double helical structure of the DNA was actually given by the Rosalind Franklin, but nowhere her name is mentioned. Okay, now what the next property they said is, these strands are held together by the hydrogen bonds. Okay, two strands which are running in antiparallel direction are held together by the hydrogen bonds. Okay, and purine always pairs with the pyrimidines. Okay, so this was the point which was said by the Watson and Crick also. And adenine pairs with the thymine, while guanine base pairs with the cytosine. Okay, so adenine forms two hydrogen bonds and pairs with the thymine, while guanine forms three hydrogen bonds with the cytosine and base pairs with the cytosine. Okay, so this base pairing, okay, gives the some sort of enhanced stability to the antiparallel strands. Okay, so this also involved in the promoting the strength of the both strands. Okay, so adenine pairs with the thymine with 2 hydrogen bonds, while guanine and cytosine pairs with the 3 hydrogen bonds. Okay, guanine and cytosine are much more stronger bonds, okay, than adenine and thymine. So the DNA which is rich in the guanine bases is actually a strong DNA and has the high melting point. Okay, the DNA which is rich in the adenine bases or thymine bases, that is a very weakened DNA, or it might have a very low melting point temperature. Okay, now, so a lot of scientists, question the Watson and Crick, why only the adenine base pairs with the thymine, why not this adenine base pairs with the cytosine? Okay, so the major reason, or the adenine base pairs with the guanine. Okay, the major reason is the distance between the two bases. Okay, so adenine and thymine base pairs, when adenine base pairs with the thymine, it forms certain distance. Okay, between the adenine and thymine we have some distance between them. Okay, the same distance is also observed between the guanine and cytosine. Okay, the same distance is present in both base pairings. Okay, so because of that distance, this adenine is pairing with the thymine and guanine is base pairing with the cytosine. Okay, uh, yeah, so this is what the distance is. Okay, so the this adenine molecule, when it is pairing with the thymine, okay, so the distance between these two bases is what 10.5 10.85 angstroms. Okay, and the same distance is also observed between the guanine and cytosine, 10.85 angstroms. Okay, so this is why the adenine pairs with the thymine and guanine pairs with the cytosine. Okay, if we replace the bases, the distance might increase, and the strands will be quite differently arranged. Okay, not the in a linear way, not in a linear way, but some sort of distortion will be seen in the helical strands. Okay, so that is why. Okay, if you combine a purine with the purine, the strand will be the strand will be too wide, and if you combine a purine, if you base pair a pyrimidine with the pyrimidine, so the strand will be very narrow, like this. Here the hydrogen bonds will be present, and the structure will be too narrow. Okay, and if you base pair with the purine plus pyrimidine, like adenine and thymine and guanine and cytosine, so it will have some sort of same distance between the bases, and uh, and the consistent linearity will be there. Okay, consistent linearity between the strands will be there. Okay, so that is why purines will be pairing with the pyrimidines, and pyrimidines will be pairing with the purines. Okay, in all the nitrogenous bases, the nucleotides, okay, so which are involved in the formation of the nucleic acids, okay, guanine has highest number of bond pairing atoms in it. It actually forms 9 bonds. Okay, then the cytosine has that ability to form the lot of extra bonds. Okay, but usually guanine only forms 3 hydrogen bonds with the cytosine, not 9 atoms are involved in the bond formation. Okay, so this is what the Watson and Crick said. There are also a lot of other properties which I'll be discussing in the forms of the DNA. The bDNA has. Okay, so the genetic information resides in one of the two strands known as the template strand. Okay, so whenever whenever an mRNA has to get transcripted from the DNA, okay, one will be acting as a template strand, and on that template strand, RNA polymerase will be binding on the template strand, and this RNA polymerase will transcript the mRNA strand. Okay, based on the template strand DNA. Okay, and the other opposite strand, which is which is running the antiparallel direction to this template strand, is known as the anti-strand strand. Okay, or the complementary strand. Okay, and the actual template strand is also called the sense strand. Okay, so this is the major difference between the template and the non-template, or the sense and the anti-sense strands. Okay, and we also have major groups and the minor groups, which will allow the lot of interacting proteins like transcription factors or the other gene regulating factors to get involved with the DNA and do their work. Okay, they actually penetrate the phosphodiester linkages and interact with the DNA bases. Okay, these grooves are meant to interact with the transcription factors or any other type of proteins. Okay, or regulatory proteins or the gene regulatory factors. Okay, so this DNA also has the major grooves. Okay, and one more interaction, one more major important thing about the nucleic acids, these nitrogenous bases are stacked upon each other. Okay, so one above the other. Okay, so what is stacking is, when you arrange one coin above the other coin like this, okay, and make this group of coins into a pile, this kind of this kind of arrangement is called as a stacking. Okay, similarly, the same kind of arrangement is also found in the nucleic acid chain. Okay, so the nitrogenous bases which forms the nucleotides. Okay, these nucleotides are arranged in the form of this stack of pile. Okay, like they are stacked above each other. Okay, so this kind of stacking involves the pi electronic interactions and gives the enormous stability to the DNA strand. Okay, so the first stability is by the stacking interactions. Okay, and then by the phosphodiester linkages, and then by the hydrogen bondings. Okay, so these are the major stabilizing forces which are present in the double helical DNA. So one is the stacking, base stacking, which we also call as the base stacking. Okay, so base stacking plays a major role in the stabilization of the DNA, double helical DNA, and then comes the phosphodiester linkage, which maintain the linearity of the DNA strand, and then comes the hydrogen bonds, which are present between the two antiparallel strands. Okay, so this also maintains the linearity of the DNA. Okay, so these are the major stabilizing forces which are involved in the formation of the double helical DNA. And as I said we have 2 nanometer or 220 angstrom diameter. The DNA, double helical DNA has a 20 angstrom diameter, and rise per helix is 34 angstroms, and rise per base pair is 3.4 angstroms. Okay, and approximately 10 base pairs to 11 base pairs are present in one helical turn. Okay, so if you also count 1 2 3 4 5 6 7 8 9, and here what 1 2 3 4 5, yeah, here we will be having one more base pair over here. Okay, at the back side. So total 10 to 11 base pairs, 10 to 11 base pairs will be present one helical turn. Okay, so one helical turn length will be 34 angstroms, and the distance between the two helical bases will be the 3.4 angstroms, and diameter will be the 20 angstroms. Okay, these are all properties given by the Watson and Crick, and they also said about the minor groove and the major groove. So in the whatever the DNA structure we are discussing, that is the B form of DNA. Okay, so that is this B form of DNA is normally present inside the cells. Okay, the general DNA which is found inside the nucleus is what? bDNA. Okay, and it has the major groove which is meant for interaction of different types of proteins and transcription factors. Okay, or the gene regulator factors. So as the additions I've discussed already. Now the conformations of the DNA. So DNA normally inside the nucleus, it exists as a B form, in the B form, the normal DNA. So when if we dry the crystals of the bDNA, okay, and if we lower the salt concentration or the salt content of the salt content in the bDNA, then we'll get a thinner molecule, which is often referred to as the ADN. Okay, so if you want to convert the bDNA into ADNA, you have to transfer the bDNA crystals, dried crystals, into the low salt content solution. Then you will get the thinner form of the bDNA, that is known as the ADN. Okay, so ADN and bDNA, the sugar group and the bases are on opposite sides of the glycosidic bond and in the anti-conformation. Okay, so there are two types of conformations for the sugar molecules. Okay, anti and syn conformations. Okay, so the ADN and bDNA contains anti-conformations. Okay, I will discuss about this anti-conformation and syn conformation, but just remember that ADN and bDNA contains the anti-conformations. Okay, and if the same bDNA, if it is transferred to a high salt concentrations of cations, okay, some nucleotides will rotate into syn conformations. Okay, and this kind of syn conformation will give different type of orientation to the bDNA, and this will convert the bDNA into Z form of DNA. Okay, so this converted form, Z form, will be in the zigzag orientation. Okay, having the syn conformations and the anti conformations. Okay, so this will result in the formation of the ZDNA. Okay, so higher salt concentrations or the higher concentration of the cations will make the bDNA into ZDNA, while the lower salt concentrations or the when you dry the bDNA crystals, that will convert into the ADN. Okay, so this is the major conversion thing. Okay, So within the cells, yeah, most of the DNA is bDNA, although regions which are rich in guanine and cytosine base pairs may assume the Z conformation, that is the zigzag conformations. Okay, and zDNA is radically different duplex structure with the two strands coiling in left-handed helices and a pronounced zigzag pattern in the phosphodiester backbone. As I said, alternating anti and syn conformations of the purines and pyrimidines will give the zigzag nature for the bDNA. So that is this converted bDNA is actually called as the zDNA, and Z is for the zigzag name. Okay, and zDNA can form when the DNA is alternating purine pyrimidine sequence such as GC sequences. When there are a lot of GC sequences, then also we can see the zigzag pattern, the zDNA patterns. Okay, the big difference is at the G nucleotides. So G nucleotides, if they are rich, they also form the triplex DNAs or the G quadruplex, G tetraplex. Okay, like when in the triplex DNA, three antiparallel strands are combined. Okay, in the tetraplex, four antiparallel strands will get combined. Okay, so these tetraplex and the triplex are very rich in the guanine nucleotides. Okay, so the ZDNA is also rich in the guanine nucleotide, and in zDNA, it has the sugar in the C3 endo conformation. I'll tell you about this, the endo and exo conformations. Okay, and this places the guanine back over the sugar ring, and in contrast to the usual anti-conformation seen in A and B DNA nucleic acid, this zDNA will have the syn conformation. Okay, if we talk about the major group in the bDNA, firstly, yeah, so in the bDNA we have a very wide major group over here, to allow the major transcription factors or any kind of proteins to easily penetrate into the DNA, to easily get attached to the attached with the DNA. Okay, while in the major group of aDNA we have very narrow major group. Okay, here, and here in the zDNA the major group is very flat. Okay, so these two A and Z DNAs actually don't allow a lot of proteins to get involved, while bDNA allows a lot of proteins to get involved for the gene regulation. Okay, these are the comparison table for A, B, Z DNAs. Okay, so if you look out the overall proportions of the DNA, first one is the aDNA is short and broad, much shorter than the bDNA, and this is actually longer and thinner, and zDNA is what elongated and the slim DNA. Okay, so when you stretch the bDNA, it will form the elongated and the slim zDNA. If you compress the bDNA, that will form the short and broad aDNA. Okay, so if you consider this as the bDNA, okay, so if you compress on both sides, if you compress the bDNA on the both sides, okay, so this will form aDNA. Okay, like this. Okay, and if you stress the bDNA in opposite direction like this, in this direction, then it will form the zDNA. Okay, so zDNA is formed when you elongate the bDNA, and much more elongated version and the slimmer version of the bDNA is what zDNA, and much more shorter version and the broader version of the bDNA is what aDNA. Okay, so this is the major differences between the ABZ and DNA, aDNA, bDNA, and zDNA. And the rise per base pair is also different in this 3 DNA forms. Okay, so aDNA has 2.3 angstroms rise per base pair, while the bDNA has 3.32 to 3.34, and zDNA has around 3.8. Okay, because it is very slim and elongated, it will have the higher rise per base pair, higher rise per the base pair. Okay, and helix packing diameter for the aDNA it is 25.5 angstrom, and for the bDNA 23.7 angstrom, and for the zDNA, because it is slim one, okay, obviously it will have the shortest packing diameter, that will be the 18.4 angstroms. Okay, and these are all the major important properties of the ABZ conformation. Okay, and we also have the major group proportions. Okay, so most of the students get confused between this major group proportions. Okay, and I have a trick to remember this major group proportion. So the trick is an extremely narrow B canal widens toward the Z line by 5 inches. Okay, so if you remember the tricks, if you remember this trick, okay, so it is very easy to remember the major group proportions. Okay, Now, what does the trick says? Okay, an extremely narrow B canal widens towards the Z line by 5 inches. Okay, so this A, the first A, represents the ADNA. Okay, and the extremely narrow represents the major groove of the ADN. Okay, and the B, the second B, which is the highlighted B, talks about the BDNA. Okay, so this BDNA what wide. Okay, and the Z indicates the ZDNA, and the second letter F, we have the preceding letter F. Okay, so this F indicates the flat. Okay, so that means ADNA is extremely narrow and BDNA is wide and ZDNA is flat. Okay, the major groove of the ABZ DNAs are in these forms. Okay, so if you remember the trick, this will be very easy. Okay, and glycosyl bond formation is what anti and anti-H C and the syn H. Okay, so the anti-H C is what pyrimidines. Okay, so pyrimidines will have anti-conformation while purines will have the syn conformation. Okay, but in both cases ADN and BDNA, that will be the anti-conformation. Okay, so what is an anti and what is a syn, that is discussed here. Okay, so the furanose ring or the ribose ring, which is present in the deoxyribose sugar, okay, if is actually a non-planar ring. Okay, so that means this non-planarity, type of non-planarity is is called as a puckering. Okay, so due to this puckering, not all the atoms lie in the same plane, but some atoms will differ out from the plane, and may exist as endo or exo conformations. Okay, so what are these conformations, we'll see. So furanose ring of the ribose or deoxyribose if non-planar, and this planarity is termed as a puckering. Okay, so individual nucleotides in solution can adopt many different conformations. For example, deoxyribose sugar can bend and twist in many different ways. So two of the most stable conformations are shown in the right side, like C2 endo and the C3 endo. So these are the major conformations which are present in the nucleic acid chains. Okay, now the C2 endo conformation allows the maximum separation between the oxygen atoms and at the 5' position to 3' position. Okay, and while the C3 endo conformation will be more compact and it will form the tighter helices. Okay, that means the sugar in the sugar puckering, okay, in the sugar puckering, what happens is out of 5 atoms which are present in the ribose sugar, okay, 4 will be in the same plane. Okay, so they are arranged in the same plane, and one atom will always be in the out of the plane. Okay, so that means it distorts the planarity of the molecule. Okay, so due to this distortion, the ribose will have the non-planarity. Okay, and that atom, it might be the C2 carbon 2 of the ribose sugar or carbon 3 of the ribose sugar. Okay, so carbon 2, it may exist as in the exoform or the endoform. That means inside endoform will be the along the plane. Okay, and the exoform will be the outside the plane. Okay, so when this carbon 2 is present in the exoform, that will be in the along that will be this carbon will be along the planar atoms. Okay, so however the atoms in the ribose atom are arranged in the same plane, the same way the carbon 2 which is present in the exoform will be in the same plane. Okay, but if the C2 carbon is situated outside, okay, so if the position of the carbon 2 is in endo conformation, so then it will be in the out of the plane. Okay, so then it will be coming out of the plane, and it will distort the structure of the ribose sugar. That will result in the non-planarity. Okay, so C2 endo conformation will allow the maximum separation between oxygen atom in the 5' and the 3' positions. Okay, and the C3 endo will be seen in the carbon third carbon. Okay, so there is also two types of conformations, exo and endo for this carbon 3, and C3 exo will actually promote the maximum separation between the oxygen atoms. Okay, but C3 endo conformation will make the helix to be more compact and the more tight. Okay, so the conformation in the BDNA and ADNA and ZDNA is different. Okay, so in the BDNA we have C2 endo and C3 exo conformation. That means maximum separation and the maximum interaction is possible in the bDNA. While in the aDNA, we have C3 endo and C2 exo, which makes the strand more compact. That is why the aDNA is what? Shorter and the broader. Okay. And this aDNA is much more compact because of the C3 endo and the C2 exo conformation. Okay. And if you talk about the zDNA, the pyrimidines of the zDNA has the C2 endo and C3 exo conformation, while the purines of the zDNA has the C3 endo and C2 exo conformation. Okay, so this type of unusual conformations will give the zigzag orientation for the DNA strand. Okay, so that is why the zDNA is named as a zigzag DNA. Okay, so this is what the C3 endo and C2 endo conformation. Okay, so when we talk about the anti-enzyme, the orientation of the N-glycosidic bond. Okay, so if the purine is located alongside of this new ribose sugar, that is called as the syn position. Okay, and if the same purine or the pyrimidine ring is located opposite side to the ribose sugar, that is called as the anti conformation. Okay, given here in the paragraph, in addition to the sugar precursors and bond rotations around the backbone, there are also many conformations of the bonds base related to sugar. The two extreme conformations involve rotation of the beta and glycosidic bond shown in these figures. Okay, so in the anti-conformation left side, the base is rotated so that it is far from the sugar as possible. Okay, that means quite opposite direction. Okay, the sugar will be in one direction, and the nitrogenous base will be in the opposite direction. Okay, so this kind of arrangement is known as the anti-conformation. While if the both nitrogenous base and the sugar, if they are in the same direction, that is called as the syn conformation. Okay, so the anti syn conformation, anti and syn conformation also gives a lot of unusual properties to the DNAs. Okay, like majority of the zDNAs will have the syn conformation at pyrimidines, and purines have the anti conformation. Okay, that is what I have given here. So this zDNA has the anti-conformation at the cytosine, okay, in the pyrimidines, okay, and will have the syn conformation at the G guanosine, that is the purines. Okay, and so most of the scientists, after knowing about the structure of the DNA based on the Watson and Crick model, so they try to figure, they have tried to figure out the properties of the nitrogenous bases, or the properties of the sugar molecules. Okay, or they want to know the conformations of the nucleotides, how they are arranged in the double helical form, the double helical strands of the DNA. But whenever the scientists try to build a model of the DNA before the Watson and Crick, they failed. Even the Linus Pauling, who also tried, who was a great chemist, who also tried constructing the double helical model of the DNA, also failed, because the conformations of the double helical strand DNA. Okay, so what are these conformations and how these conformations are changed? That is due to the seven horizontal angles of which determine the conformation of one nucleotide chain. Okay, so each nucleotide generally has 7 horizontal angles. Okay, so one is alpha, beta, gamma, delta, epsilon, and zeta. Okay, so these are the 7 horizontal angles. We also have another horizontal angle, that is a chi horizontal angle, that is present between the base and nitrogen atom, base and sugar molecule. Okay, so the N-glycosidic bond will also show some angle movement. Okay, so that will result in the one extra horizontal strain. Okay, horizontal angle. Okay, so yeah, chi, the horizontal angle describes the N-glycosidic bond and varies between purines and pyrimidines. Okay, so these types of horizontal angles will give the lot of different conformation to the nucleotides, and these two nucleotides won't stay in a consistent position. Okay, so they change rapidly. Okay, so this kind of rapid movement will have not allowed the lot of big scientists to figure out the double stranded helical form of the DNA. Okay, so these are the about the horizontal angles. So first horizontal angle alpha is present between the phosphate molecule and the oxygen atom, phosphate atom and the oxygen atom, and the second beta is present between the oxygen atom and the carbon 5 atom of this ribose sugar, and the third gamma is present between the C5 and the C4, so carbon 5 and the carbon 4, and the delta is present between the carbon 4 and the carbon 3, and epsilon is present between the carbon 3 and oxygen 3. Okay, and zeta is present between the oxygen 3 and the phosphate of preceding nucleotide, okay, whatever the nucleotide that is present between the initial nucleotide. So between that phosphate and the oxygen we have the another torsional angle, okay, and the chi torsional angle is present between the nitrogenous base and the pentose sugar, okay. So this point you have to remember, okay. So they might ask you in the national level entrances what type of torsional angle is seen between the base and the pentose sugar, okay. So this point you have to remember. And apart from the properties of this different types of conformation and the properties of the nitrogenous bases, there is also one more extra property known as the oxygen base pairings, okay. Oxygen base pairing is quite different from the Watson and Crick base pairing. So Watson and Crick base pairing, what it says is, adenine base pairs with the thymine with 2 hydrogen bonds, and guanine base pairs with the thymine, guanine base pair with the cytosine with 3 hydrogen bonds, okay. And adenine thymine, when they base pair, they involve 6, 1 and 4, 3 atoms, okay. So these atoms of the ring are involved in the formation of the hydrogen bonds, while when guanine base pair with the cytosine, okay, so 6th atom, 4th atom and the 2nd atom, okay, and 4th atom, 3rd atom and the 2nd atom are involved in the formation of the hydrogen bonds, okay. So these are the atom numbers which are involved in the formation of the hydrogen bonds in case of Watson and Crick base pairing, okay. But what this oxygen base pairing tells is there are certain conditions inside the nucleus, like when there is a low pH conditions, or whenever different types of regions of the DNA, like guanine rich regions of the DNA, want to base pair with another strands, okay. So at that time, different types of base pairing will be seen. So that those kind of base pairings is called as the oxygen base pairings, okay. And these kind of oxygen base pairings majorly seen in certain sequence motifs, like when there is a guanine rich sequence, or when there is a low pH conditions inside the nucleus, or in those conditions, oxygen base pairing will be seen, okay. And normally the triplex DNAs and the tetraplex DNAs, which I'll be discussing in front, they also contain the oxygen base pairing, okay. So these unusual base pairing property of the nucleotides will change the complete orientation of the nucleic acid strand, okay. As I said, Watson and Crick base pairing involves the sixth atom, first atom, and the 4th atom and 3rd atom of the adenine and thymine, okay. And the guanine and cytosine involves 6, 1, 2, 4, 3, 2 atoms of the rings, okay. But the oxygen base pairing involves 6th atom and 7th atom, okay, instead of first atom which is involved in the adenine, okay. So the 7th atom is involved in the nitrogen, 7th atom is involved in the adenine, okay. And that pairs base pair with the same only, like 4 and 3, okay. So the opposite strand conformation is not changed, only the main chain conformation is changed, okay. And if you look at this guanine, the 6th atom and the 7th atom is involved in the formation of the hydrogen bond, while in the cytosine, the same fourth atom and the 3rd atom is involved in the hydrogen bond, okay. So due to this unusual base pairing, the orientation of the nitrogenous bases will get changed, okay, into the syn conformation, okay. So this type of syn conformation will give the different structure to this entire nucleoside strand, and make the strand to look as kinked, okay, like it will distort the linearity, okay, if the DNA strand in this way, okay. So this kind of oxygen base pairs will make the DNA strand to look like this, okay. They will produce some kind of distortion in between the strands, okay. So this is called as the oxygen base pairing. And as I've said you, in the oxygen base pairing, nitrogen 7 is involved in the formation of hydrogen bonds rather than the nitrogen 1, okay. And this kind of oxygen base pairings also results in a different angle between the glycosidic bonds and a smaller distance between the carbon 1 atoms of the paired bases, okay. So because of this, 10.4 angstrom distance is reduced to 8.5 angstroms, okay. So the distance also get reduced and the linearity get disturbed, okay. As I said you, the triple helix DNA or the tetraplex helix DNA, they contain the oxygen base pairings, okay. So now the tertiary structure of the DNAs. So what we have discussed till now is the secondary structure of the DNA. Now one more level of the DNA is the tertiary structure of the DNA. So these tertiary structures are majorly formed when there is a recombination undergoing, or when there are transcription factors getting bonded to the DNA's major groove. Okay, so or when there is a quadruplex DNA or the triplex DNA. Okay, so this kind of condition will result in the formation of the next level structure, that is a tertiary structure of the DNA. Okay, and most of the tertiary structures are due to the formation of the G tetraplexes. Okay, so ends of the telomeres, when they overcome the end replication problem. Okay, so telomere ends contains a lot of guanine-rich sequences. Okay, like this, and these guanine-rich sequences folds into the DNA strands. Okay, so to form the tetraplex DNA. Okay, so this kind of tetraplex DNA also involves the oxygen base pairings, and this type of structure comes under the tertiary structures of the DNA. Okay, and we also have A tracks between the spacer regions. Okay, like this. So we have a adenine rich bases between some sort of a spacer region. Okay, spacer region in the sense normal DNA. Okay, and between these spacer regions we'll be having a lot of adenine rich sequences that will also produce some sort of distortion. Okay, so this also result in the formation of the tertiary structure. Okay, which will bend the DNA. Okay, so these kind of structures or unusualities in the DNA will make the DNA to be in the tertiary form. Okay, first one is the curvature of the double helix over several bases results in the bend DNA. So that means when the DNA has A tracks between the spacer regions. Okay, continuous A tracks between the spacer regions will bend the DNA, and a DNA kink causes the change in strand orientation in an otherwise linear double DNA strand. Okay, so the kink is due to formation of any transcription factor or any gene regulated factor. At that time these factors actually bend the DNA. Okay, or produce some kink in the DNA. Okay, and in the holiday junctions during the recombination events when the strands of the DNAs, both the DNAs gets exchanged, at that time we'll be seeing the tertiary structure of the DNA. Okay, so this is actually the holiday junction, so where the exchange of the genetic material is actually happening. So at that time, there is a structure of the DNA is formed. Okay, and in the quadruplex DNA, where the four guanine sequences or the four antiparallel strands of the DNA are involved in formation of one helical DNA. Okay, so that is the quadruplex DNA. Okay, what we have here is a bent DNA. First one, bent DNA is due to presence of the 8 tracks adenine rich sequences between the normal spacer regions. Okay, and we also have a lot of other bent DNAs in nature. So first one is the IHF DNA. So this IHF DNA is normally called as the integration host factor DNA. So when this integration host factor complexes with the DNA, and it produces some bent in the DNA. Okay, so this roughly bent structure is due to binding of this integration host factor. Okay, so this factor contains a lot of proline residues, and these proline residues are these proline residues intercalates into the DNA strand, and they destack the base pairs. Okay, destacking in a sense they decreases the interaction between the base pairs. Okay, so that the strand will be in the strand will look like in the bent form. Okay, so the strand will be in the bent form. Okay, one example is what IHF integrating host factor and DNA complex. Okay, so this complex will produce the bent DNA. And next one is what the holiday junction, as I've said you. So when the T7 endonuclease is bound to the holiday junctions, it also produces the kink in the DNA, so that this kink is about 80 degrees. Okay, and this junction will be open to for the further exchange of the genetic materials, or for further recombination events. Okay, so these are the example for the bent DNA, which are which are unusual things, but these kind of properties majorly we'll be seeing for sometimes, like for seconds to for 10 seconds to 20 seconds time. Okay, then after that everything will be get normalized. Okay, and certain anti-tumor drugs such as cisplatin. Okay, so they also produce the bent DNA structure. Okay, so when it produces the bent DNA structure, a lot of gene regulated proteins can bind to the DNA and stop the DNA replication, or can control the can control the gene regulation. Okay, so this kind of bent DNAs are actually useful, even though they are unusual properties of the DNA. Okay, unusual structures of the DNA. Okay, so we have 6 different types of the tertiary forms, okay, or non-bDNA forms. Okay, so these non-bDNA forms are of 6 types. Okay, first one is the bent DNA, and then we have the slip DNA. So, and then we have the G quadruplex, which is a helical DNA with 4 anti-parallel strands. Okay, and then we have the cruciform DNA, which is normally observed in the tRNA molecules, or some sort of junctions in the DNAs. Okay, so when there are a lot of junctions in the DNAs, you can observe this cruciform DNA. And triple helical DNA, or the HDNA, is majorly due to the presence of oxygen base pairings, or at the end of the G telomerases. Okay, telomerase will normally have the G quadruplexes, and some telomere ends will also have the triplex DNAs. Okay, and ZDNA is normally the slim and elongated DNA, the unusual zigzag DNA. Okay, so these are the major 6 types of DNAs. Okay, so which are strongly screw stretches. Okay, and they also contain a lot of homopurines or the homopyrimidine sequences. Okay, the bent DNA contains the A tracks between spacer regions. Okay, so that produces the bent form. Okay, that makes the DNA to be in the bent form. Okay, and we also have some sort of repeating structures between the spacer regions, which make the slipple strand. Okay, and we also have loops in the DNA, which makes the G quadruplexes. That is called as the G4 motifs. Okay, so these are the different types of tertiary structures of the DNA. Okay, you just have to remember the major important points about the DNA. So now what we have is the triple helical strand of the triple helical stranded DNA, which is also called the HDNA, and this DNA is mainly due to presence of the extra guanine-rich sequence. Okay, when extra guanine-rich sequence comes and joins in in between the helical strands, and this guanine-rich sequence is actually base paired with the both helical strands. Okay, oxygen base pairing, and they form the triple helical structure. Okay, so this triple helical strand DNA formation may occur due to additional hydrogen bonds between the bases. Thus, a thymine can selectively form 2 oxygen base pairs to the adenine AT base pair to form the TAT. Okay, so thymine or guanine-rich sequences. Okay, and as I've said, guanine can form almost 9 bonds, 9 hydrogen bonds, and similarly, the cytosine, adenine, and thymine also has ability to form extra bonds. Okay, so they can modulate it to form the oxygen base pairings in the triple helical DNA. Okay, and this triple helical DNA can have number of biological consequences, including transcription repression, or site specific mutagenesis, or recombination, or even the regulating the genetic gene expression. Okay, it can also involve in the regulating the gene expressions. Okay, and triple helical DNA is mainly due to the presence of homopyridine and homopyrimidine regions. Okay, at that regions of a supercoiled DNA, we can see this HDN, or the triple helical strand of the DNA. Okay, which is involved in the regulation of the gene expressions. Okay, and then we have then we have is the 4-stranded DNA, or we also call as the G tetraplex. Okay, because it is very rich in the guanosynucleotides. Okay, so if we look at the structure of the tetraplex DNA, triplex DNA. So this is a double helical DNA, and we have one more strand, third strand. Okay, so this third strand intercalates between this double helical form. Okay, in this way to form the triple helix. Okay, so this triple helix may be in the parallel orientation, like the orientation of the third strand will be in the same direction as the complementary or the sense or antisense strand. Okay, and if the third strand is in the antiparallel direction to this blue strand, to the second strand, that will be in the antiparallel orientation binding. Okay, so this kind of binding will result in the triplex DNA. Okay, so mostly thymine rich sequences, or the homopurine or the homopyrimidine rich sequences will form the triple helical DNA. Okay, the 4-stranded DNA is also a similar way, similar DNA, just like this triplex DNA. Okay, and this contains a monovalent cation, sodium or potassium cations, in the middle of the triplex loop. Okay, so there are 4 antiparallel 4-stranded DNA structures which are defined as G tetraplexes. Actually, 4 strands which run in antiparallel directions combines to form a G tetraplex. In between the G tetraplex, it includes a monovalent cation. Okay, and these tetraplexes mainly involve the oxygen base pairings for their stability. Okay, so this is known as the 4-stranded DNA or G tetraplex. Okay, and G tetraplexes normally forms at the telomerase ends. Okay, so when the telomere telomers, when they overcome the DNA replication end replication problem of the DNA, these telomers actually contain a lot of guanine-rich sequences, and these guanine-rich sequences actually penetrate into the double helical strand and form the G tetraplexes. Okay, and these telomers, which are ends of the chromosomes, are rich in guanine, and they regulate the telomerase activity. Okay, so they don't allow the telomerase to cut the DNA sequences at the ends of the chromosomes. Okay, and in recent years, telomeres have become the target for the anti-cancer therapies because they regulate the growth of the cancer cells. Okay, if whenever you call a cancer cell, that means this cell is rapidly growing without any gene regulation activities happening. Okay, so if you want to control the cancer cell growth, then you have to cut the telomerases or telomere ends. Okay, by activating the telomerase activity. Okay, so when you can do that, when you can activate the telomerase activity, or when you can increase the telomerase activity in the cancer cells, then you can control the cancer growth. Okay, and a lot of cancer cells tend to exhibit the uh, extended telomeres, extended lengths of the telomeres. Okay, extended lengths of the chromosomes in their ends. Okay, so that is why they are actually fast growing in nature. Okay, and then G tetraplexes have also been implicated in the recombination of the immunoglobulin genes. Okay, like in the VDJ recombination, because when we do this kind of recombination events, they cause the increased levels of the immune responses or the development of specific immune responses. Okay, so this kind of implication or this kind of VDJ recombination implication in the G tetraplexes DNS will develop the specific kind of immune responses. Okay, so this is about the G tetraplex DNA, and this also has the parallel and anti-parallel nature. Okay, parallel G quartets or the G tetraplexes will have the 4 strands running in the same direction, that is in the parallel direction, while in the anti-parallel G tetraplex, we will be having 2 double helical DNA strands which will be running in the anti-parallel directions. Okay, and they form the uh, G tetraplex. Okay, so one will go in this direction and this strand will go in the opposite direction like this will form the anti-parallel G tetraplex. Okay, so this is about the structure and forms and conformations of the DNA. So in the next class we'll be talking about the kinetic properties and kinetic classes of the DNAs. Okay, so